Thank you for that lovely introduction, Ariel. I don't think of myself as the foremost anything, but it's nice to imagine yourself otherwise. All right. So I'm calling this talk Building a Brain Map, Ethical Issues on the Frontier of Neuroscience. I thought this was a little bit of a haughty title, but I'll let y'all be the judge. I'll say a little bit more about who I am, a little bit more about the specifics of my projects. Um, so I'm an assistant professor in the Bioethics and Humanities Department in the School of Med here at University of Washington. I'm the neuroethics lead on the Human Mammalian Brain Atlas project. We're going to talk a lot about that in a few minutes. So you'll know what that means when we get there. I think it's important to let you know up front, this is one of my disclaimers. I'm trained as a philosopher, PhD in philosophy, postdoc in a philosophy department, so on and so forth. But I earned my PhD doing work in a laboratory. So I was considered for a long time an embedded humanist. Um, I wouldn't call myself an embedded philosopher, an embedded ethicist. Um, but I think it's important to recognize that lots of different kinds of humanists.
We cannot hear you. We cannot hear you.
That's weird. Okay, I got it.
It's okay now?
It was not muted a second ago. Okay.
Yes,
Okay. Um, yeah, so I was embedded in an electrical engineering laboratory. And there I studied the user experiences of neurotechnology. But more recently, I've shifted focus from neurotechnology, which is like a downstream trans, tech transfer, um, yeah, a tech transfer space um, where I'm working with end users who are part of patient populations that are receiving some kind of therapeutic benefit or assistive benefit from the technologies that we want to produce. Um, and I've switched to more of an upstream space where we're thinking about the neurotechnology of, I mean, not the neurotech, the technologies of doing basic neuroscientific research and being responsible in laboratories as we do wet lab research and that kind of thing. I still do both. I still do the neurotechnology stuff. Before I go any further, I think it's important for me to say I have no financial conflicts of interest to declare. I am funded by NIH by the grace of God, apparently. I don't know how that worked out, but it turns out that the current administration hasn't taken aim at ethics itself yet. When that day comes, we'll have a lot of really deep issues to consider. But we'll talk a little bit more about that also. I'm National Science Foundation funded. Ted Cruz did put me on one of his hit lists. One of my projects mentions gender. Ooh. I've done work funded by the MacArthur Foundation, and I've given unpaid talks. Oh, it is being weird.
You're tall.
Yeah, I'll take a couple steps back. Hi, Al. Um, I've given unpaid talks to Meta, Facebook Reality Labs, Microsoft Research, IBM. I'm currently a board member for the International Neuroethics Society. I am cultivating relationships with these companies for them to give that foundation money so that ethics is important, y'all. Okay, but yeah, these are my disclosures. I thought I would start off though by giving a brief overview of bioethics as a concept, zoom in on neuroethics as a concept, and then some of the sub-concepts within neuroethics so that everybody's on the same page about this ethics stuff a little bit. How many of you have a humanist background? Ah, two of you. What's yours? Psychology? Very cool. And you? Okay, great. Awesome. I love history. My sister was a history major. How about you? Yeah. Oh, okay. Yeah, that's like very overlapping. Yeah, very, very cool. Okay, but no philosophers or like language studies or like okay, yeah. Okay, good. Good, good. So this is useful. This is going to be useful for you. Okay, so what is bioethics, right? So bioethics, I take it as the study of moral and ethical issues that arise from medical domains, usually surrounding very difficult medical cases. And these are common and abundant. Um, and ethicists usually hone in on these cases to figure out best practices, principles, lessons learned. Um, and many of the best tools that come out of bioethical analysis are connected to things like the Hippocratic Oath. So this principle of do no harm comes out of the Hippocratic Oath. Um, the Nuremberg Code, which is the international community's response to Nazi Germany's experimentation on humans. I wouldn't call it experimentation, honestly. It's just forced experimentation. The Belmont Report and Beecham and Childress's Principles of Biomedical Ethics, I take these as very connected documents and milestones in bioethics history. These are a response to a number of controversies around the turn of the century. More like in the 30s and 40s, we're talking the Tuskegee experiments, which went a lot longer than you would think, right? So, mostly the Tuskegee experiments got a lot of public attention in the 60s and 70s. And the Belmont Report is a response to public awareness of the Tuskegee syphilis experiments. You get four principles out of the Belmont Report and Beecham and Childress's principles. We get beneficence, so you ought to do good and prevent harm. Non-maleficence, so you should not do harm to people. Um, Respect for autonomy, right? So we shouldn't coerce people. We should respect people's autonomous choices. Um, and justice. And in the Belmont Report, this gets couched as one ought to treat equals equally. But what does that mean? It usually means something like we need to distribute resources to people, we need to distribute burdens to people in a way that feels fair. So one group of disadvantaged people shouldn't shoulder the burden for everyone else. And one group of elites should not receive all of the benefits of biomedical research. And these principles sort of uh, they're conceived in the concept of doing biomedical research, but they apply to medical practice as well. I know a lot of people lean into the Hippocratic Oath, but these principles are there. And of course, these are not the only resources we have available in bioethics. I'm only scratching the surface to bring us to the same page. Um, so what is neuroethics, right? So Adina Roski's a pretty prominent bioethicist has given us two ways of thinking about neuroethics. And so she makes a distinction between the neuroscience of ethics and the ethics of neuroscience. We'll talk a little bit about each, but these two are starting to overlap quite a bit these days, which I think is really interesting. The neuroscience of ethics is an attempt to figure out how ethics work, how morality works at the level of neuroscience, right? So what structures of the brain are implicated in us doing the right thing? Um, on the other side, the ethics of neuroscience is about responsible conduct or the social and ethical, legal implications of the work that we do as neuroscientists, right? So how is my research project going to impact the community? Who gets hurt through the actual process of doing the work? Um, What kinds of protections need to be in place? How do I become the best neuroscientist possible? That's kind of a virtue ethics way of thinking about it, but interesting. So I think this is a good distinction. But we'll look at both separately as well. So the ethics of neuroscientific research. This is what I mostly do, but I'm doing more work in the other area as well. So we know that the foundational and basic neuroscientific research can transform clinical practice in lots of different fields like neurology, neuropathology, psychiatry, psychology, on and on and on. But to do this kind of work, well, a lot of neuroscientists would say to make an omelet, you got to crack some eggs. I would prefer no eggs be cracked, especially if that's my skull. So this requires invasive studies, sensitive data, sensitive specimens. We're talking animal studies, we're talking invasive human studies, human subjects research. And so human subjects, well, all subjects are vulnerable to physical harm, exploitation, and misrepresentation. And so we have to figure out what the safeguards are to do that work responsibly. On the other side, we have the neuroscience of ethics, which has its own precarities, right? So we're trying to figure out why people are moral or immoral. And underneath it all, we're trying to figure out ways to intervene on the immoral people, right? Seems like a really hotbed topic, right? Immorality is rampant. I keep thinking of this weird movie, Little Nicky, what's it called? Adam Sandler's movie, right? Where two devils kind of take over New York, right? And then at some point they have, I think the Archdiocese of New York say sin is in. That's kind of what it feels like to be in this country right now. So like immorality is like rampant. I just don't get it. But yeah, they haven't taken aim at me yet, so we're okay. Um, but in trying to figure out why people are moral or immoral, we have to figure out a lot of different things. We have to figure out what behaviors we're going to call immoral. We have to figure out what the mechanisms, what the pathways, the neural pathways are, what the neurocircuitry is of immorality and morality. Spoiler, some people think it's like oxytocin. Some people think that's the reason we're moral is because we have oxytocin in our systems. It's not the case. It's very simplistic, very reductionistic, actually. And then we have to investigate these behaviors using neural imaging, observation, sometimes invasive measures, right? We're talking like neural implants. But who are we gonna approach? Hey, immoral person, let me study your brain. No, we're doing these studies on prisoners. That's how this works in a lot of different countries, including so like in Central and South America, lots of neuroscientists are doing this kind of work on prison populations. And we're doing it in the United States too. I just don't think we have the funding for it as much. But trying to figure out where people are trying to figure out the neural correlates of immoral behavior, but they're embedding a lot of biases about what counts as immoral behavior. So any kind of violence is considered immoral. Anger is considered immoral. Where does that leave me? I'm angry all the time. So, and I have been called immoral in high school by white teachers. That's a thing. Oh wait, I don't know if I'm supposed to talk about that. Uh-oh, I might get canceled. It's funny how that word flipped. Like, who's doing the canceling now? Okay. Um, but yeah, all of that said, what is this weird brain atlas, brain map stuff that I'm working on now? Um, so so the National Institutes of Health have a brain initiative, and this is an Obama era institute or initiative that is supposed to bring together a lot of different neuroscientific projects and fund them well. And we've been eating good since Obama put this in place. And so I guess I'm not supposed to say that either. But one of the big flagship projects in the Brain Initiative is BICAD. So this stands for Brain Initiative Cell Atlas Network. And this is NIH's investment in multimodal research. So this is an attempt to create a reference atlas of brain cell types across different kinds of analysis. So we're doing genomic analysis, transcriptomic analysis, so RNA analysis, spatial analysis, functional analysis. And the whole point is to figure out what kinds of cells there are in the brain, where those cells are located, and how they're connected with other cells in the brain, as well as where they're located in specific people's brains, because we know that the brain has regions and they are specialized, but we also know that that specialization happens in different people in different ways, right? So it's paramount that we have brain donors, brain donors that have undergone functional MRI. And so we know that such and such brain cell was in such and such place and performed such and such function and has such and such properties. Really cool. But we're also doing this across species, right? So, and we'll talk a little bit more about that. But uh, the project that I work with is working across 4 species, um, uh, rodent, marmoset, macaque monkey, and human. And that's an attempt to make sure that we understand how different species have different cells in different locations, but we know what the commonalities are. So if you've ever heard of these problems with translating between animal models and human models, this is supposed to overcome that. If we know each brain intimately, then we can transfer knowledge about rodents more easily to humans. And this will help drug studies, neurology studies, so on and so forth, right? And of course, they're working across developmental timelines. So we're talking about, infants who have lost their lives, unfortunately, but we can use their brains to understand infant brains or fetal brains, right? So I work within the Human Mammalian Brain Atlas project. This is one of the funded projects from BICAN. I'll just call it HMBA from here on out. It's a flagship project within BICAN. So I'm the lead ethicist on a flagship in a flagship in a vanguard, which is weird. It sort of fell in my lap, and I don't know why. This is a collaboration between UW's Brain Lab, which has a brain donor network, and Paul Allen's Center for Brain Science. So we just call that uh, uh, I guess they call themselves ABES, right? But I call it the Allen Center. So we use specimens from human donors that are donated through the medical examiner's office here in King County. The Brain Lab has a deep relationship with them, and they've been allowing the Brain Lab to reach out to families of bereaved families of people who end up at the medical examiner's office, and they collect brain specimens, brain donations that way. And so this also is supposed to be buttressed by a new palliative care and hospice care donor program where we would approach people who are end of life, terminally ill, but without any brain issues, and ask them if they want to be brain donors. And if they do, we give them that functional imaging and we collect their brain donation when they pass away. And we combine all that data and we have the best possible brain map, which I think is a wonderful prospect and fits a lot of people's end of life goals really well when we can reach the audiences that we want to. This also built on Alan's outreach. They've been doing brain map type things. They're very interested in the connectome. So this idea that brain cells are connected in certain ways And it's extremely important that we understand those connections. So how does the HMBA work? We get a donor and they pass away. They end up in the medical examiner's office, and the medical examiner allows us to do a number of things. First, we do a scan of the person's brain while it's in their body. We call that a cadaveric scan, cadaveric MRI. And then the specimen is harvested, I don't like that word. It's collected and it's subjected to another scan. And we call that a fresh MRI, right? And all of the information that's collected before is used to guide a parcellation process. The brain is sliced into thin slices, imaged, and subjected to really detailed genetic analyses, of which I barely understand. I'm a philosopher, guys. But what I can tell you is that they're using RNA RNA-coded dyes to make the cells fluoresce. And they use a very, very beautiful, I've seen it in person, scanner to identify all the cell types in parcellated tissue. And so I think it's really interesting. And this produces a 3D explorable model. But how do we select the brains, right? Of course, here's just another way that we're thinking about the process of collecting brain donations. At some point, when we receive a brain donation, and we perform the scans, we do a number of different tests. We do interviews with their families, the donors' families. We access their clinical records. We do infectious disease testing. We do toxicology. We do slab parcellations and see what the data does. And we test for what we call RIN. So this is RNA integrity. Since a lot of our work requires RNA to be intact, RNA integrity is paramount. And we do neuropathology. And this is probably one of the most important things that we do, is we check to see if the brain has been damaged somehow. So if a person has signs of Alzheimer's and they don't know it, that shows up in the brain in the form of plaques. And those plaques make it very difficult to do the analyses that we want to do, make it impossible. And so after that, we end up, after that information collection process, we look at that data together as a group. And I'm in those meetings as an ethicist, but there are so many different people in the room, and they're trying to pick brains. And so we call this a consensus review. So I want to say a little bit more about what we're selecting for. We're trying to find and sort these brains into quartiles, right? And we really want the top 25% of brains in terms of tissue and in terms of, or at least I know this is a bias of theirs, neurotypicality, right? And we'll talk about that concept later. That's one of the big issues, right? So first quartile donations are used first, and for the most important studies, we're producing an entire brain map from those brains. The team started calling these golden brains. It's kind of weird. I tried to make a pamphlet for prospective donors that had a golden brain on it. It looked very weird. I did not like it. Lower quartile donations are used if there are unforeseen problems with the first quartile donations, because we're really just making a projection as to whether or not we'll be able to use the tissue. And all donations are supposed to be used for population studies that are meant to study cell type and anatomical differences across human population generally. So the idea for the main studies, the most important studies, is that this brain is going to be used to do a very comprehensive sampling of cells and analysis of all the cells eventually. Um, but but we can do lower resolution sampling and get a sense of how brains differ across social and cultural demographics, across patient populations, so on and so forth. And so that was supposed to be a really big part of this project, but we received a bunch of budget cuts. We received uh, a lot of just straight up budget cuts, but also new restrictions. So we had international partners, and those have been cut out of NIH entirely. You can't have international partners anymore because I think the rationale is we don't want taxpayer dollars, American taxpayer dollars, going overseas or something like that. Usually that money went to bringing people over and draining people's brains. And we had great colleagues in Germany, Italy. Yeah, we had a lot of great colleagues. Spain, I believe. And now we don't have those colleagues. They're still our colleagues. It's not like they disappeared or something. But yeah, the population studies have also been cut, because we didn't have what they gave us a 10% cut and then another 15% cut. So that was the only part that the golden brain studies were the ones that were deemed essential, right? Um, so let's say more about this brain selection consensus committee. We meet once a month. People from across HMBA are welcome to join us. So from the people who, oh yeah, go for it. Oh, okay. Feel free to ask questions if you want. Um, but yeah, so we meet once a month. Um, Researchers and technicians from across the consortium are invited to join us. There are usually about 10 to 20 voting members at these meetings. We look at all of the data that we collect about the donation and the donor and their family. Um, compiled into small case reports. That is really a streamlined process at this point. And really all we do is we consider the case. Dirk Keen, one of the PIs, he leads us through the case. And we have a conversation about the merits of each case, and we vote for what quartile to put them in. It's a pretty straightforward process. But lots of complications emerge, right? So here are three cases. You tell me how you feel about them. So in case A, there's a 18-year-old Hispanic who died accidentally as a result of the toxic effects of fentanyl. So there's a concern for polysubstance dependence because the toxicology screens were positive for fentanyl, and the urine analysis was positive for other drugs like cocaine and meth. The tissue quality was very high in terms of this RNA integrity, and there was no neuropathology, so no plaques or anything like that. But everyone was concerned about the polysubstance abuse, right? So case B, we have a 32-year-old white non-Hispanic female who died as a result of fentanyl and ethanol and intoxication. So they took fentanyl, drank a lot, and they passed away. They had a long-standing history of psychiatric issues, so ADHD, depression, anxiety, and substance abuse. Tissue quality was high, though. Are you starting to see the pattern and the controversy? Case C, a 50-year-old white guy. He served in the military. Uh, he had a benign clinical history, no evidence of neurological or psychiatric disorders. negative for infectious disease or toxicology. Um, he only had caffeine, sleeping pills, and weed in his system. That's it. Tissue quality is high, and neuropath is negative, right? So, but still, this case is controversial. We had a long conversation about whether or not serving in the military would cause or make you vulnerable to concussion. And one of the people on the call who served in the military as a medic said it's all but certain that this person suffered a concussion, even though there was no evidence of it. So, controversies. Our biases about what lifestyles cause what tissue quality degradations were beginning to drive the selection process. In all of these cases, the tissue quality was high. And there are, and I'm not putting these on slides, lots of cases where the tissue quality seemed high, but as soon as we got into the analysis, it was not high. There were lots of problems. And some of these cases that where a person died of fentanyl overdose or were identified for substance use, their tissue was better. Go for it.
These like the cases where they are like reporting psychiatric history or whatever or all of those things, like isn't it very possible that you have a domain brain that the person did have symptoms of depression or anxiety, but like no reported psychiatric history or like they never sought treatment for that, so it's not on the record. Is that not like sometimes concerning? How much is it based off of what is actually reported and so?
I actually think this is one of the, this is a great question. Yeah, sure. So hopefully I summarized this right. So what happens when a donor shows up and they have no history of depression or some sort of psychiatric condition, but maybe they do, maybe they were depressed, maybe they did have ADHD, maybe they did have anxiety, so on and so forth. It's just undiagnosed or undertreated, or those records were lost, right? I call this an epistemic gap. And these epistemic gaps are everywhere, right? These are knowledge gaps. And I think it was one of the turning points in my engagement with this group when I had to tell the group, hey, absence of evidence is not evidence of absence. And everyone went, huh? And I'm like, yeah, just because we don't have the evidence that they were depressed doesn't mean they weren't. And we know that a lot of people keep their mental health issues to themselves. So case C, you know, what older white man who served in the military wants to seek out therapy? This is a really difficult problem in psychiatric practice and psychological therapeutic practice. Getting people to get therapy, to have that on their record. And a lot of people would take that to the grave that they feel depressed, right? So what sense does it make to say people should seek out mental health treatment, but then in their end of life, when they're seeking out ways to contribute to science in their last years on this earth, no, you can't participate in this because you have this history of depression that we told you that you should have so you could seek out care, right? It's a conundrum. It's not good. And that's why a lot of things have changed in our consensus committee. The culture of our consensus committee, our process hasn't changed, but our culture has. I should say, we have a neuroethics team. That's the whole point of this team, is to consider issues like these. My student, Zoe Hale, whose picture is right next to mine, she sits on the consensus committee meetings, and she's wonderful. We also have an advisory board made up of wonderful neuroethicists, including Adina Roskies, who's one of the founders of neuroethics. But yeah, here are the issues that come out of HMBA. So we ran into the issue of achieving a heterogeneous sample. I'm using the word heterogeneous now. I would have said representative. That's one of the dirty words. So we got to be careful. It's heterogeneous now. um, uh, Exclusion of non-neurotypical donors, right? Along the same lines that we were just talking about. Recruitment practices, relationships with families, data sharing and analysis and presentation, including responsible uses of AI. My group is a little cagey about this. They're like, how does this have anything to do with, or I don't know how it relates. It must, but I don't know how to do it. And we're still working on that. Maybe at uh, the next meeting of this group, I can give updates along those lines. But we've mostly been thinking about recruitment issues and follow-up issues. But we will talk a little bit about data sharing and analysis. So how do we achieve a heterogeneous sample, right? So initial problems are the golden brain group is very small. We're talking 6 people. That has been expanded to about 15. But it's still a very small sample. How do you achieve representation? How do you achieve heterogeneity, especially in Seattle? It's a very white place. And how do you represent the even smaller groups like American Indians and Alaska Natives? And as we saw with the earlier cases, different people have different health outcomes depending on their social positionality, right? So if you are lower income, if you're a woman, if you're a person of color, if you're queer, those identities make you vulnerable to different forces and make prosperity a little bit harder to access. And so those show up in the reasons why people end up in the medical examiner's office. That's the reason why people overdose, right? That's the reason why people commit suicide, right? So on and so forth. Mistrust and distrust will make it really hard for people to want to contribute to research. We've had Black donor families who've specifically called out Tuskegee. They're worried about it. Representation is hard to achieve in this city, of course. And as we were talking about before, and this is NIH's policy, they don't want people who've overdosed, people who have psychiatric disorders. And they don't want them because they want to study those conditions. So they also want to study brains that don't have those conditions. But I don't know if there are brains that don't have those conditions. I think all of us have issues. So this idea of neurotypicality drives a lot of national policy about what brain donations get used for what project. There's an intuition baked in here that donors have to be neurotypical to ensure high tissue quality. I've already called that into question. And that donors have to be neurotypical so that these samples or donations can serve as a control for future studies. So once we have the control, we can study people with target conditions. But what do we even mean by neurotypical? I think there are 2 different ways of thinking about neurotypicality. There's a brain scientific model of it. So neurotypical means you don't have a disorder. But on a social model, it means that you're a part of a community of medical practice, of solidarity, of shared experiences, of having to fight against a medical system or get a diagnosis. That's what people who are ADHD or autistic on the spectrum will report and find communion in. And so there's this distinction between neurotypicality and neurodivergence, neurodiversity, and neurominorities, right? So a lot of people will posit that people with neural differences are minoritized in this country and globally, of course. And so they would say that they constitute a neurominority. I think it's cool to put neuro behind anything, and it makes it more legitimate, right? Um, so I think we're encoding, these intuitions encode a bias into our data, right? So if we exclude for depression, anxiety, trauma, drug use, we're going to exclude everybody, everybody. No one's going to count as typical. So we have to make some concessions if we're going to use these intuitions, right? And experiences, life experiences don't necessarily correspond with downstream neuroanatomical differences, cell differences, any of those differences that we want to study. So what we arrived at is this idea that HMBA, the goal is to produce a reference. We're trying to produce a reference of the highest possible quality. We're just trying to say these cells are here. We're not trying to say this brain is perfect, right? Because no such brain exists. And also, why would we want to study that brain? That brain hasn't lived, right? Okay. So this is less important now because, well, I guess it's important for us. NIH wanted us to have, a representative population. This is pre-Trump, of course. Now I think they want all white. But they wanted ethnic and racial diversity in these proportions, right? But that would mean we would never have any Native Americans ever. There are issues there. They wanted veterans. That's part of the list of minoritized groups produced by NIH. I don't think the current administration cares about veterans at all. They closed down the VA, so. And yes, neurotypical donors were preferred. But for us, we decided to favor allocation over exclusion. We're part of a network of a lot of different projects. So if a brain can be used, we're going to use it because it's a precious, precious gift that a donor gives to us, that a family gives to us. We include subjects from the following groups whenever possible to support heterogeneity so that we don't exclude populations inadvertently. So when somebody commits suicide, when they overdose, when they have a history of depression or anxiety, as far as we can, we'll include those groups. We cannot include Alzheimer's, Parkinson's, or any other neurological condition like traumatic brain injury that would impede the research itself. The main driver of exclusion from our study is tissue quality. If we can't do our work, then we don't want the brain. But we can use it in another study on Alzheimer's. Really cool. So we use quota-based sampling. We're aiming for a quarter from these groups, but a quarter of each of these groups. Native Americans are a special case because we need tribal consent to use those brains, and we don't have it. So I've pretty much told them we have to build that pipeline, we have to build those relationships, otherwise we're just not going to be able to do that. And that leads into this idea that we need to build relationships with marginalized groups, right, and communities. But we have to go beyond that. So even if we're not working with those groups directly, we have to do a good job of recognizing that those groups exist, representing them well, and paying reparations to the groups that have been harmed in the past. Otherwise, we can't do research with those groups ever. Whoa, I'm always hitting that thing. I left a dirty word in there. So justice, equity, diversity, inclusion efforts have to extend into data management, analysis, and product development as well, even if no one participates in our study, even if they all say no. In terms of relationships with families, families could really benefit from seeing the impact of a loved one's donation. There's a family called the Dahl family. There was a boy fell out of a second story window when he was young, 10 years old, ended up in the hospital. in critical condition, really, really bad concussion, really bad traumatic brain injury, and made a full recovery. Did all the things that you would expect a kid to do if they didn't have a traumatic brain injury. But eventually, after college, uh, you know, young adult Matthew Dahl, he developed a deep, deep depression. But it wasn't like the depression that a lot of us feel, like a neurochemistry imbalance that could be settled by antidepressants. No, he said that something was fundamentally wrong with his brain. And he would tell his mother that something was wrong with his brain. And he eventually took his own life. And this was relatively recently, I think 2019. And so Matthew's parents, Bob and Linda, donated his brain to one of our projects. And they found the neurocorrelates of traumatic brain injury that would cause depression. And it gave them a lot of clarity. And that clarity, they really had an impact on them. They're deeply involved in the research still. Well, Bob recently passed away. They donated his brain to research as well. And so, but Linda is still involved in our research. I just saw her at one of our meetings a couple of months ago. So wonderful stuff. Big possible benefits. And marginalized people would definitely welcome the opportunity, at least some of them, to represent their communities in this research. They know they've been excluded. They want to be included. And so, but the problem is that these contributions, they're being used for genetic analysis. And genetic analysis is precarious, right? If we handle the data incorrectly, draw conclusions that misrepresent the family or the community that the family comes from, we can do serious harm. This is the problem that Henrietta Lacks's family. How many of you know about Henrietta Lacks? Okay, cool. Currently, the Lacks family is fighting a fight to keep people from doing genetic sequencing on their on Henrietta's cells and publishing it in journals. And so they've already come up with a way for scientists to access those data in a secure way that doesn't involve just putting like the text in the PDF and publishing it. This was a collaboration between them and NIH. I don't know where that collaboration is going to go under this new administration. A lot of things have changed, right? But yeah, these data are precarious, right? And they're very intimate. And so some families may not want to be exposed that way. And cultural and religious beliefs might be inadvertently violated. There's a great paper called Will I Go to Heaven If They Take My Brain? And it explores this issue of people's different cultural beliefs. Where in that paper, there's a Catholic saying, will my body resurrect at the day of judgment if my brain is in a laboratory somewhere? And his conclusion was, God would make it possible. He's omnipotent. So I think this is really interesting. I get giddy about these. I used to be a philosopher of religion for like 20 minutes. But yeah, I was really into it. Then I realized that one of the biggest groups of philosophers of religion are rock climbers, and I'm not climbing rocks. No, I'm afraid of heights. That's the real reason I stopped. But yeah, so we have to be sensitive. Our recruitment methods have to be respectful to the cultures that we want to recruit, recruit from. And our obligations go beyond recruitment. We just need to engage with the groups that want information about what we do. Public perceptions of science are at an all-time low. Our ratings are low. We need to boost those ratings. We remember the non-maleficence and beneficence principles we considered earlier, that we shouldn't harm, that we should cause good. We have to be able to take no for an answer and for a community to still benefit from our research. That's been my line for as long as I've been part of this project. There's an ongoing case I think is extremely important. This connection that we have with families is extremely important. So there's a person who signed up to our hospice and palliative care program. They had terminal cancer, but they were highly secretive about their choice to become a donor for us. They didn't really think their family would approve of it. When they passed away, the family didn't know how to contact the brain lab team. They didn't even know she was part of the study. Her living next to kin contacted us eventually, but the donor brain was not collected in time. So we really can't use it for our study. And now we're scrambling to figure out who can use this tissue within our BICAN consortium and our network of projects. And we're trying to learn from this experience. We're trying to figure out what language we can give to people to give to their families to approach this issue of brain donation, right? And that's frustrating, right? And so this is difficult. I'm starting to think, what is this group getting out of this conversation? And here's a bone. I'll throw you a bone. It's important to realize that these data are going to be made public and available to everybody, right? This is again, I say this, these are vulnerable, these are sensitive data from vulnerable people who made a really gracious decision to give their brains to us. And so imaging, multiomics, and limited metadata will be available through a public explorer. People will be able to access levels of these data and apply for deeper levels of access to the data. And of course, these data are going to be fed into machine learning algorithms. They're going to be fed into artificial intelligence. We're already using artificial intelligence to identify the cell types, right? And that raises a whole other set of problems. Most paramount to the group is this idea that data need to be fair, so findable, accessible, interoperable, reproducible. The whole goal of the project is to produce fair data that you can just type in a command and have access to, which is wonderful, accelerates process, right? I mean, progress. But data analyses need to use fate or fact principles, right? So that's fairness, accountability, transparency, and ethics, right? And so, yeah, ethics is a big one to stick in an acronym like that. I don't know. I'm not a fan, but I'm glad to be acknowledged. I'm glad for the shout out. And so we need to be transparent in how we collect the data. We need to be fair in how we distribute the data, and we need to be accountable to downstream harms caused by making the data available. And the best way to do that is upstream. When we're looking downstream at the possibilities, put the safeguards in place, right? We need to think about the interaction between these principles, right? Fate and fair, they sort of overlap with one another. So for example, when a tribal nation here in the States wants control of the data, does that mean we still get to do open access to data? No, we don't. We have to limit that access according to the needs of the community that's making the data available, right? And so fate and fair may come into conflict with one another. It's important to remember fair principles are not necessarily for moral consideration. Fate principles are moral, right? So we have to be careful, right? And finally, and I'll close out with this, fixed data ontologies clash with social ontologies, right? And we saw this with this idea of neurotypicality, right? We have these scientific categories, and we're trying to find neural correlates, even more scientific categories. But are neurodivergent people going to agree with those categories, or the implications of using those categories on what lives they have to live downstream, right? So the clash is there, right? And that's where I stop. We don't have time for questions, but questions? Yeah, thanks.
We'll circle the topic if people have questions. Let's do kind of a couple questions.
Okay. Go for it.
MRI, person passed away and the brain is inside of the bone,
Yes.
and they wanna,
Yes.
uh, when the pressure is. Question, you're a philosopher, so why did you go and wrote?
I know a little bit about this actually, right? So, oh yeah, okay. So you do we do a cadaveric MRI and then we do an MRI after the brain has been extracted. The pressure changes the morphology of the brain. How do we correlate those data together? And we've excluded brains because that process wasn't done right. And so the process is pretty interesting. You know, it's we have an experimental setup that that correlates the two together. There are some really fancy algorithms. Um, uh, there's one of our colleagues, Eugenio, I always forget his last name, came up with a really interesting method of making sure that the morphology doesn't change too much, and doing the imaging in a way that correlates well. But if the cadaveric MRI isn't done, that becomes extremely difficult. But yeah, there's a bucket and a plate and an elaborate camera setup, and it does a really good job. And the algorithms correct the distortions in morphology pretty well. They also do like perspective parcellation, not parcellation. Yeah, I guess yeah, it's parcellation. So it color codes everything for us, and they actually do that for before consensus committee meetings. So if it wasn't, if they ran into trouble doing it, we'll just say, oh well, this is a consideration against it because it's going to make the rest of our analyses more difficult. But yeah, all that to say, fancy algorithms do a really good job. Yeah, no problem. Thanks.
You could talk about that a little bit.
Yeah.
I'm thinking about if like certain demographics are more open to, donation and like then you deal with like the bias of the data set or are you representing the population?
Yeah. So I guess the question is, are there some populations that are more enthusiastic about being donors?
And also like are there considerations to reduce bias?
Yeah, and yeah, how do we reduce bias? It's really difficult because yes, there are some populations that are much more enthusiastic. On the neurotech side, people were much more likely to get brain implants when they were older white guys because they wanted to be Chuck Yeager. And they've said that to me before. They want to be the person who goes first. They want to be the pioneers. Another colleague of mine, Sarah Gehring in the philosophy department, chair of the philosophy department, she has a project that's wrapping up right now called Supporting Brain Pioneers. I think that's the language. But yeah, brain pioneers being these people who use neuroprosthetic limbs and things like that for the sake of assistive rehabilitative goals, largely white guys, because they want to be first. As in like they want to break the sound barrier or something. And I think that's awesome. And I appreciate their enthusiasm and their contributions. But on the other side, a lot of marginalized people don't want to be first. Like that's part of our culture. No, not me, not yet. I'll wait until version 8, right? And that's what I get. That's a response I get from my family when I tell them about my research. They're like, I don't want to. So how do we overcome this bias that just comes as a result of people's interests and their very real concerns? We have to be open with them and we have to build relationships. So right now, me and a colleague within Allen Brain, Jimena Garcia, We're reaching out to different community organizations and leading community events so as to do some public outreach about our research, to raise awareness and also to get input if they want to give input, both in Latin American communities and Black communities, because those are the communities most accessible to me in Jimena. But yeah, the key is communication, and the key is language right and facilitating conversations that are open and honest. And I think that is going to do a lot. But my metric isn't, oh, more people in the study. My metric is better educational benefit for the community, right? That's my metric. So yeah, I hope that was helpful. Cool.
Okay, we close here. Thanks
Cool.
again, Tim, for that, and we'll be back with the next talk at 30 past the hour, so the coffee break will be a little bit shorter. Thanks again, Tim.
Thank you.